Okay, well, it is 10 a.m., so I think we'll get started. So, hello everyone. My name is Nissa Krull. I'm a PhD candidate in the Murphy Lab at UIC, and I'm happy to see a few familiar faces from our last workshop. Thank you for returning. And if this is your first time joining us for an IDBac demo, welcome. We want to thank you all for taking the time to be here with us today. We're very excited to show you all the new features that we have implemented on the web platform and the database. Now, off the top, I would like to highlight the IDBac Workshop team. It has truly been a group effort to make this happen. We have Dr. Laura Sanchez at UC Santa Cruz. We have Dr. Ming Wang at UC Riverside. And I want to highlight two very important people that are in the audience here today. We have Michael Strobel. He is in the Wang Lab, and he has put in tremendous efforts in creating the Web IDBac platform. We would not be where we are today without his tremendous efforts in creating this user interface. So thank you to him. And Robbie Shepard is also on this call. He's a part of the Sanchez Lab, and he has been a fantastic help from the start, early beta testing, documentation review, and he's been working on a very exciting extension to IDBac that we're hoping to make public within roughly the next year or so. So both Michael and Robbie are on this call. They're going to be in the background. They will help keep track of some questions, maybe flag me down if I miss something in the chat while I'm talking. So again, thanks to them for their work and for the work that they're going to do today. All right, and just a note, I believe all of you probably received the notification when you signed on. We are recording this session so that we can post this later on our YouTube so that you and others can reference these demonstrations at a later time. Okay. So to give you a broad overview of the workshop, we are going to begin by discussing the current status of the database. We will walk through how to deposit into the database, and then I'll give you a demonstration on protein small molecule analysis. And I will show you how to access the documentation that, again, can help you as you hopefully jump on and explore our platform. And we'll highlight a few interesting things that are coming down the pipeline. I also would like to add, please feel free at any point during this presentation to interject with questions. You can add them in the chat, or you can feel free to email me at any point that questions or comments arise. Again, we are very reliant on your feedback and greatly appreciate you taking the time to do so. Okay, so jumping into it, the IDBac.public database. Our objective here is to create a robust and taxonomically diverse multi-top MS protein database that is user populated and open access to the broad scientific community. We have been able to start the baseline volume of reference strains by generous contributions from collaborators from various different institutions. We've highlighted a few here just at the bottom. Oops, lost one of my AirPods. My apologies. We have the Dutroux Institute at the University of Chicago that has contributed microbiome data. We have the Puny Lab at the University of Wisconsin, who's contributing micromonosporosae, the NASA Jet Propulsion Laboratory Planetary Protection Program, and they've contributed quite a few diverse genera to the database, and we're hoping that they are going to contribute several hundred more in the coming months. And we have the Metcalf Lab in conjunction with the USDA that has allowed us to incorporate a large collection of streptomyces from the NRL collection. And we have the Linnington Lab at Simon Fraser University that is contributing Burkholderia species. And we are hoping that we can add you to the bottom of this list. Again, to create a robust and taxonomically diverse database, we are going to rely heavily on users to help us populate. So again, we said this in the email as well, we more than welcome contributions, and I'd be happy to discuss any contributions that you may have. So I want to give you a little tour of how to walk through our database. If you go to idbac.org, you will land on this page. You can see a table right off the bat. You have strain names, you have things like GenBank Ascension. These are all columns that we ask for when you in the metadata table that we'll get into in the next couple slides. But these are all metadata columns that we ask for to help give users of the database more information to work from. If you are interested, you can click on one of the strains and scroll down here and you can visualize a spectrum. You can scroll down to see additional data, for example, the taxonomy, the 16S sequence, if this was submitted as 16S verified. And you can also toggle what columns that you would like to see. If you want a very long list and you want to scroll, you can click everything. Or if you're, for example, just interested in the full taxonomy, you can deselect a lot of these and shorten your list to highlight the columns that you are most interested in. You also have the option to export this into an Excel document. So this is the current status. As I mentioned, we've had contributions. So we are up around 1,000 strains, reference spectra at this point. We are hoping to at least triple that in the coming months. And we're excited to see this grow as the community starts populating it as well. Do we have any questions on the database itself before we move on to how to deposit strains? Seeing something. Let's see. And we will move on to how to deposit strains into the database. I'm going to spend quite a few moments for database depositions and also for analysis metadata. Metadata is a very crucial part of the process of submitting workflows. So I would like to highlight a few things. You can find these sheets in our documentation. They're linked to a Google Drive. And we also have instruction tabs on those Excel workbooks, or the Google Sheets, pardon. There's an instruction sheet that'll tell you what each column is and what we're requesting and why, but I'm going to highlight them here too. Everything in blue is crucial. You have to submit all of these columns when you are depositing a strain into the database. Now, the tricky part here is this zone in the center. This is also required, but you don't have to require every single one of these. So we are asking, again, for genetically verified spectra strains to submit to the database. So we are asking for either a GenBank Ascension number, an NCBI tax ID, or if you have 16S taxonomy that you have not yet submitted to GenBank, for example, you can provide that. You can blast your sequence, you can write down the taxonomy here in this column, but we do ask that you provide the 16S sequence so that users can run blast analysis in the future if they so choose. Now, once you have this metadata template, and as you can see, similar is a reflection of the database. We have, again, the isolate source, longitude, latitude. You can add your own comments if you would like at the end here. As much information as you can provide is very beneficial for users going forward. So whatever you can share is much appreciated. So once you have your metadata sheet filled out, and you also have MZML files. All of our workflows require MZML. Again, the documentation can walk you through some conversions, and I'll discuss a workshop that we'll have coming back that discuss how to get to the point of MZML. But once you have MZML files and your metadata sheet, you can access the GMPS2 landing page. And to find the workflows, you click here. And as you can see, there's a long list, so it's easier to start typing in IDBac, and you'll see the workflows that are specific to IDBac. Since we are discussing IDBac depositions, we're going to go here. And there's just a few little sections here to fill out. Now, this can be anything you'd like. I personally like to highlight the dates and then maybe the collection that I'm depositing, whatever is going to make sense to you later on if you're trying to look for the task in the future. And then anytime I click on one of these green buttons, you will be given this pop-out window where you can access, oh, I think we gotta put people mute. You will get this pop-up window and you will access any of the files that you have submitted to the file browser. Now I can go down here. I highly recommend, I mean, you can see I have many folders. I've been doing many different trials for the Web IDBac. Hopefully for you all, there'll just be one or two here. But I highly, highly recommend organizing your folders in the way that's going to be easy for you to identify quickly when you're running these different workflows through GMPS2. So deposition here, I have my deposition metadata. I'm going to click that and then add to the metadata file. And then I have my folder that has my MZML. I'm going to click that and add into the input spectra folder. Now, this right here is very important, the dry run mode. For this workshop, I'm going to keep this to yes because I'm not going to submit these strains right now. But if you are ready to go, you want to deposit strains directly into the database, make sure that dry run mode is clicked to no. Clicks to yes, you won't see it show up on idbac.org, and we'll have to clone and rerun the workflow. So again, I'm just going to click dry run mode, and it'll take me to this page. You can pay attention to the status area. It will either tell you queued, running, or done. And once it's done, you can give it maybe 10 to 20 minutes, depending on the traffic on the server. Give it 10 to 20 minutes, and then you can go back and reference the idbac.org, and you should see your strain populate. One way that I think is very easy to look up, a task that I've done, you can look for this task ID right here. If you copy that and you go to the task column, to find it on this new toggle. If I input this here, I should be able to search the deposition that I just did, or I can search by maybe the culture collection. There's several options to try to kind of expedite you finding your strains in the database. All right. Do we have questions on database depositions? And I keep referencing the documentation. Again, I'll show you this at the end, but there are many steps like uploading your files into a file browser. All of that's covered in the documentation, but I'm happy to answer any questions about that moving forward as well. If there are no questions about database depositions, we're going to move on to the analyses demos. Again, I'm going to linger on the metadata for a little bit longer. As you can see, we have a second metadata sheet that is quite similar to that first one, but there are a couple of very important differences here. We have 2 extra columns on the protein and small molecule metadata sheet, or on the documentation, it'll say analysis metadata. These 2 columns incorporate the ability to run small molecule data as well. It's important to note you can run protein data without small molecule. You can have a full protein analysis run without small molecule, or you can run them together. And to show you an example of a metadata sheet filled out, I have my strain name, I have my file name. This represents the protein, and then the small molecule file name, which represents the small molecule. Now you can notice here, they are identical, and that's okay because I'm going to submit them into separate folders when I am submitting my workflow, which we'll see in a couple steps here. So don't worry if they are the same name, but ultimately all of these labels are entirely up to the user. You can label them as you wish, just as long as whatever's written here reflects the file names that are in the file browser. You're good to go. Now, the other factor here is the blank file name. This is important again for the small molecule. We recommend that you take on your MALDI-TOF plate that you have a media and matrix control, and you are going to add that into the file browser as well. We will subtract that from your small molecule spectra for the metabolite association networks. And the rest of the sheet is identical to the protein metadata, or the deposition metadata sheets. More information you can add that's useful for yourself. We are not incorporating small molecule into the database this time, so really this sheet is up to you on what you would like to analyze with your data. So once we have that filled out, I showed you how to access the workflows before, so to save us a little bit of time, I linked us to the IDBac analysis workflow directly. But again, you can find this going through workflows and searching for IDBac. There's the same. You can label this however you see fit. Oftentimes, I will put analysis. The pages that you receive, the landing pages from here will also note analysis, but when I'm running down the list, I found it useful to add in deposition or analysis into my job title. And then same as the deposition, we have a lot of green buttons over here. As you can see, there are quite a few more files that we need to submit for the analysis portion. So I'm going to ignore my deposition folders because I've already done that. That's not part of my analysis, but I'm instead going to focus on these down here. I have the input protein. You can either go this way or this way. I prefer to go down my list as it's listed here. Media control, I'm going to find my control folder. I'm going to add metadata to the metadata, protein, small molecule. And then when I'm done adding my files, I hit close, and you can double check here. If you're a little weary, if you're like me and have many different folders to click through, you can always come to this page and double check that the right files are in the right folders. I'm going to scroll down here to the parameters. There are two in particular that I think are useful for you to pay attention to. Now, you can change the protein database search mass range, the lower and upper ranges. That's entirely up to you. But this is defaulted at 2,000 and 20,000 to start. And you can also look at the distance metric. You can choose from presence, cosine, or Euclidean, whichever you prefer. I'm going to leave this at cosine for right now. And then this is the protein database search distance threshold. It is defaulted to 5, but again, this is a score that you can manipulate yourself. And we are talking distance here. If I'm using cosine, 0 is identical and 1 is dissimilar. So maybe I'm going to turn this to 0.7 to show me isolates that have a distance up to 0.7 or spectrum. My apologies. After that, I'm going to hit submit workflow. And again, you have this landing page. You have your status that hopefully says running for you right off the bat. And these workflows can be generally quite quick, but this is highly dependent on how many users are using GMPS2 at that time, how large your data set is, or how high you set that database threshold. The higher that you set this, it requires the workflow to search through more and more spectra, so it can take a little bit more time to process your data. Now, there are a couple options when this is done. For the sake of time, I'm going to go to a task that has already been done. Once it's done, you have your analysis options, and this is where we get into the fun analysis part of the workshop. We have the database search summary tool, and this is pretty quick to explain. This is allowing you to visualize your data and database distances in a table format. We have here the query file name. So whatever streams that you just submitted for analysis, this is in this column. We have the database strain names, the culture collection it comes from, the we have the taxonomy, and then we have the distance. And you can toggle this. So if I want to see the distances that are closest to 0, I can toggle this. Or if I want to search by my query file name, I have that option as well. We will be editing this page a little bit. We might remove a couple of these columns to just clean it up and make it a little bit more concise for you all as you're navigating it. But this is a nice way to look in a table format of all the strains in the database and the distances, all the hits from the database and their distances. Now, the one that we're going to spend a lot more time on is the downstream interactive IDBac analysis. Now, I also want to note, if you go to analysis.idbac.org, I think we have that, if we can throw that in the chat. We've added it into our emails as well, but we'll make it very accessible to you. If you go to analysis.idbac.org, you will see what I'm looking at. We have a sample data set for you. So if you are not quite ready to analyze your own data, but you'd like to play around with the platform, you have we have a handful of strains to work with from the start. So we have several different tools over here to the left-hand side. We are going to go through each one of these, and we're going to start with the protein dendrogram. But one thing that we have hoped to do for you is to provide many different analysis options so that you can walk onto our platform and you can put in data and come up with new research questions. I'm excited. Sorry, do you have a question? No, okay. We have many different options on each of these tabs, and I think it could be a little bit overwhelming and a little bit dry if you watch me click through every single drop down and every single tab. I think it's the best way to get a feel for this platform is to really jump on, and we highly encourage you to use it, give us feedback on what works, what you think should change. So instead of clicking through everything today, I am going to present this as if I have research questions. I'm going to present to you research questions that I personally have had with several of my projects. So we'll start at the top here. This is kind of general. We have the IDBac logo, fantastic. This is that GMPS2 ID that is also found on your page. And this is, again, useful. We hope that you don't run into problems. But if you do run into a problem and you email me saying, Nissa, I got this error message. If you incorporate this, this will allow us to very quickly try to remedy whatever the error is. We have the workflow parameters. This is essentially everything that you chose from that workshop, the last page when you were submitting your strains for analysis. And then you have potentially an error warning pop up here. Now, we have worked on the wording for this. We want to allow users to know if there's something wrong with any of their files. So these error messages right now say scan 2 contains no peaks. If I were just analyzing protein data and not small molecule, I would only have one of these warnings. But since I have small molecule and protein files, it's telling me that my scan 2, which is representing my replicates on my plate. So my second well of the one strain, it has no peaks. It's a flat line spectrum. Now, I run replicates of 5 when I'm running MALDI, so I'm comfortable moving forward because that means I have 4 replicates that I can work with in the data set that I'm about to look at. But this is entirely up to you. You can decide if you want to proceed, if you want to remove strains from your analysis. Hopefully, these file warnings can give you a heads up before you start digging into your data too deeply. If you don't want to look at this anymore, you want to hide this, you can click or declick the show warnings, and that will disappear for a little while. The last very kind of specific thing I want to show you is this metadata tab. If you are running only protein, this only works for protein, not small molecule at this point. If you're running protein and you did not want to submit metadata in the previous step, you just wanted to throw your strain IDs and your MZML files into the workflow and look at the dendrogram, that's okay. You can do that, and you can drag and drop a CSV or XLSX file here to incorporate your metadata into the interactive platform. Okay, so now let's start going down the list. I'm going to collapse all of these to make it a little bit cleaner, again, so that we don't get too overwhelmed with too many options to start. I am going to start as a researcher that I just have many different environmental isolates, and I'm interested in seeing just a general composition. I want to see how strains are clustering within my dendrogram. So I am going to choose my clustering method. I can choose average, I can choose complete. I'm going to choose whatever I see fit. And then I can also work with this little slider bar here that will color code clusters on dendrogram cut height. This is just useful for me if I want a quick look at my dendrogram. I can set this to 0.7, and this is going to say anything at 0.7 with a parent node height at 0.7 or below is going to be color-coded differently. Anything that the parent node height is above 0.7 is going to be this blue color. So this is useful for just quick looks at your dendrogram. I've enjoyed this when analyzing data, especially quite large data sets. This is a little small one here. This can come in handy for kind of quick qualitative analysis. And maybe, again, I have these environmental isolates. Maybe I'm a microbial ecologist, and I am interested in metadata associated with my strains. Now you can see here I have my strain ID, and if I would like to, I can add some metadata feature next to it. Let's say temperature. If I click temperature, I'm going to see the temperature populate next to my strain ID. And again, or maybe I had submitted 16S taxonomy onto my analysis metadata sheet, and I can check taxonomy for all of these strains. But another way that we can visualize metadata that I tend to prefer is through a scatter plot next to the dendrogram. So I might be interested in the isolate source of my strains. Let's say this is marine sponge swab 1 and freshwater swab 2. Whatever your sample sources are, you can look to start seeing if you see any trends. Like here, I see from swab 1, I have this whole cluster showing up. We're very excited about having this feature, and you can add as many metadata columns as you would like. You can stack them, and then you can also take PNG images of whatever you have plotted next to your dendrogram. And I might be interested too, if I have these unknown environmental isolates and I'm wanting to see if it hits with anything in the database, I might notice something in my dendrogram listed DB results, and that's going to indicate a database hit. And I can choose what I would like to see about that database hit, like the taxonomy, which could be useful to me. Now, if you have, again, a much larger data set, and as we grow our database composition, we anticipate that you might have more hits, and perhaps you're interested in only looking for one specific taxonomy. Maybe you are looking for a Citrobacter, for example. So you only want to see Citrobacter populated in your dendrogram. You have that option to curate what database hits show up and how many, essentially. And this tab is kind of the last one. You can select isolates to hide from the dendrogram if maybe you forgot to remove a calibrant well, or there's an isolate that you're not happy with the spectrum after you've QC'd. You can remove it from the dendrogram so it's not impacting the clustering. You can also add dendrogram cut height to help you visualize data and display dendrogram distances as well. Now back to thinking as an environmental researcher potentially, or maybe a drug discovery researcher, I might want to look through my data set and decrease the redundancy in my collection. So I might look at some strains like this, 3A053 and A091. These are clustering quite close together on this dendrogram. The distance is 0.12. They look highly similar. So perhaps I don't want to spend the time and the money to sequence both of these strains, but I'm going to do a little bit more analysis before I make that conclusion. I can go over to plot spectra, and I can plot a single individual spectrum like this, or I can go find those two strains that I was just referencing and create a mirror plot that is showing me, oh, there are quite a few peaks that are matching here. I think it's highly likely that these are very similar bacteria. And if I want to send this to my PI, I can label this anything. Ooh, caps lock is on. I can label this whatever I would like, and I can take a PNG. I will have this nice image that I can pop into an email for presentations or further analysis. Now I'm actually going to jump because I want to stick with this thread of maybe I'm trying to decrease redundancy. One other thing that I can do, if I really am really interested in seeing how similar or dissimilar these bacteria are, particularly as a drug discovery researcher, I can move on to a metabolite association network or small molecule platform or option on our platform, where I can visualize our protein data as the large nodes and the small molecule data, M over Z's, as these smaller nodes. If I would like to, I can populate this by strain. So I could try to search just those two strains that I was interested in. Let's give it a second to think. You can always, while you're running this platform, especially on the Metabolite Association page, it can take a few seconds longer to process whatever you're doing. But you can highlight these strains and see, oh, look, they're not sharing anything right now. Maybe I can manipulate the relative intensity to see if that changes the metabolite association network, or I can change the replicate frequency threshold. And this is saying how many replicates did this specific M over Z show up. I can manipulate both of these slider bars, and that will change the small molecule association network down here, as you can see. Now, again, there are so many different options that we've labeled on here. We've had a lot of fun adding in options, like you can change colors and you can change the node shapes. Many different options for you. We're hoping that you can use this for your own analysis, but then also present and be able to present your data in the way that you would like it to, instead of having to move to another resource to visualize your data. So we have, again, there's a lot of similarities here to the IDBac software, small molecule analysis and metabolite association networks. But we do have one other aspect that is very interesting here, which is the protein or the small molecule heat map. See if it wants to quote. I got to add more strains here. We can add several strains and scroll down. And as you can see, we are looking at a small molecule heat map. And again, it's taking a second, but here we go. We have, this is essentially another way for you to visualize your small molecule data. We love the MANs for describing these data and being able to communicate about our data, but we also want to offer something like a protein, or I keep saying protein, my apologies. We also want to provide heat maps as an alternative way to analyze your data. So here I can look at these two strains and say, oh, they share this specific M over Z and this one. You can remove the dendrogram if you so choose. And you can choose up here, you can filter how many M over Zs are represented with so many strains. So if I set this, I'm saying, show me M over Zs that are associated with at least one strain. Show me M over Zs that are associated with at least two strains, etc. There doesn't seem in this data set that there are any at this intensity that are associated with more than one strain. So, any questions so far? It's gonna stay on the small molecule, but either the dendrogram page or the small molecule. It's a lot to take in, I know. All right, so if there's no questions, then we're gonna move on to the protein heat map. This is why I kept saying protein earlier. I've been doing a lot of work on this one with this heat map. Similar idea to the small molecule heat map. We wanted to be able to provide a resource for people to visualize their data, their protein M over Zs, in a way that's different than just a dendrogram. And you have several different options on how you want to build your heat map. We're modifying and working on what options we're offering here, but you have essentially 3 different options to start with. You can build a heat map based on your protein clusters. So let's say I'm interested here, and I know that these are specific. This is a specific taxa of bacteria, and I might want to know perhaps like maybe there's a biomarker in here. Maybe there's an M over Z that's consistent. So I'm going to choose cluster 3 to create my heat map. And if I scroll down, again, I get a heat map of those specific strains that I chose. I have my relative intensity scale, and I can also, again, modify the relative intensity. I can modify the minimum M over Z count. This is, again, show me M over Zs that are in at least 1, at least 2, etc. Or if I want to go a different route, I can just choose specific strains that I'm interested in. Same idea. I will have a protein heat map that is just specifically these strains that I'm interested in. Or again, back to maybe metadata trends that I'm interested in. I can create a heat map based on my isolate sources alone. So if I click isolate source, you see my swabs show up here. So that's swab 1, but I can incorporate all of them if I so wish. I can update the filters. And then both on the small molecule heat map and on this protein heat map, you have the ability to search for specific M over Zs. So if there is something, if there's an M over Z of interest or you notice a trend, you can zero in the heat map so you're not looking at every single option and really highlight what strains. So let's say I'm interested in 4197 to, let's see, 4875. I can search within this mass range and I get a much smaller, much more digestible heat map. You have the option to download the current heat map into an Excel doc, or again, you can take a PNG image of your data in this layout. Any questions on heat maps?
I had one quick question. It kind of ties into everything that you've shown with the drop-down menu where you can select, isolate source or all the different metadata options. Is that, it looks like it's not alphabetical. So is it based on the Excel spreadsheet that you upload with your metadata, like the order in the drop-down menu?
Yes. Are you talking about this page or were you referencing specifically?
It kind of applies to all of the pages you've shown so far. So yeah, in that drop-down menu where you have isolate source,
Yeah.
or in any of them, yeah, because it's kind of the
Mhm.
same, it's pulling from the same data file. Is
It
that
is. So they are they're ordered. If you see here 1, 6, 8, 9, 10, they should be in theory ordered based on whatever data you're inputting. They should be alphabetized. It gets a little different when you're working on the clusters or the metadata, other metadata categories, but the strain IDs are alphabetically listed.
Okay, awesome. So for the metadata, specifically, the order is based on how you have it in your file your Excel file that you uploaded, not alphabetical.
So So we actually have the ability, we can change, if you're talking about like this column here or this drop
Yes,
down here,
exactly.
we have the ability to change that on our end, what order all of these show up in. This is showing up in the order that we have the metadata sheet laid out. If you recall the metadata sheet that I showed as an example,
Mhm.
something like the comment was all the way at the end of the sheet. So it's just taking that format going left to right. We do have the ability though, if we get users that tell us, hey, we want to put some of these less useful metadata categories towards the end, we can absolutely do that.
Okay, cool.
But
Thank you.
sure. Any other questions? All right, if there aren't any more questions, we're going to move on to the documentation. I want to show you how you can access this. So when you sign up today, again, I will send out the YouTube link to this Zoom recording so that you are able to reference this at your leisure. But if you want a little bit more guidance as you're working through, you can go to GMPS2, the homepage. You can click, apologies, I keep clicking around too quickly on you. You can click documentation. And then you scroll down to find IDBac and you can see we have a page on database depositions, analysis. We have raw Bruker data and XML to MZML guidance. But this is constant. This is a constant update. I am tweaking little aspects of the documentation as we make improvements on the web platform, but it is a really good place to start if you are interested in either depositing strains or running your own analysis with your own data. And then let's see. And then a few exciting things that are coming up that I wanted to share with you all. Our last workshop, we had some feedback that people would like a demonstration from start to finish. Since we had so many new users, we were wanting to do kind of a broad overview, show you what's available right now. But we are going to offer another workshop next month, September 18th at 10 a.m. CST. And this would be kind of a mini workshop where we work through data conversion. So raw Bruker data, collecting my data. We can even discuss plating techniques, etc. But we can go from raw Bruker data to GMPS to IDBac workflow. We will go through the entire process instead of jumping in once you already have your converted files. So at the end of this presentation, we're going to have an exit survey where I will ask for your interest for a couple of these things on the coming soon page. If you would share with us your interest, we will send you invitations to join. We also have some exciting database updates. As I mentioned, we are constantly working on the documentation and we're always trying to optimize the platform. There'll be database peak matching improvements. We are working on just user interactions with the web platform. We're constantly making changes. And again, we really hope that you can send us feedback on how to better improve some of the user interface. And we're also going to work on some database depositions that are lined up. This is just a select few. There are many more genera that are going to be added in the coming months. Excuse me. We have Burkholderia species, Myxobacteria, Selenospora, Micromonospora, Salvaraspora, Pseudomonas, and again, many more that are going to be deposited quite soon. Several of these should be deposited within the next couple of weeks. So again, expanding our taxonomical diversity and hopefully strengthening this database little by little. And then we're also going to offer some video tutorials, adding to the documentation. We will post these YouTube sessions, but I'm also going to start working on screen shared and narrated documentation or tutorials on how to do each of our workflows and how to run each of our analyses options. Again, that's the idea where you can listen, you can pause, you can try it yourself at your own time. We hope to get that out in the next month or so. And then finally, I'm going to start offering, I tried to say host and offer at the same time. It did not work. I'm going to offer some Q&A sessions that are going to be hosted bi-weekly. This essentially will be me providing a Zoom link. And then if you have questions, say you were trying to run an analysis and you run into problems, or you would like some guidance on one of the workflows, you can drop into one of these Zoom meetings and we can discuss whatever you'd like to talk about. Again, please feel free to reach out at email anytime. You do not have to wait for a Zoom session in particular. I just want to open up another way to get a hold of us and ask your questions. I want to give a big thanks again to all of you for taking the time to be with us here today. Big thanks to all of our collaborators that have generously contributed to our database, and again to the IDBac team as a whole. And I think Chase, I didn't get to acknowledge Chase in the first slide, but Dr. Chase Clark was the creator of the IDBac software that we've been jumping off from and making the new improved version of. But I want to give him his due here. He did fantastic work on the IDBac software. We are grateful for him. And then if you'd be so kind, we have this exit survey here that Robbie has produced. It's just a few questions asking for your feedback on the workshop itself, and then asking your interest in the upcoming workshops and Q&A sessions. So with that, I will take any other questions that you have.
Nissa, Monica asks, how much data should be deposited for authorship?
That's a good question. That's a good question. We are nailing down the exact amount. I think maybe a minimum of 20 strains at this time. We want to highly encourage, even if you just have 5 strains as you go, we really, really would like your help in populating the database. But I think a minimum of 20 strains could move towards authorship.
There was also a comment earlier about receiving GNPS2 tokens. So
Let's see if I can bring up. I sent out all except for one. There was one email I received this morning, but all of the other requested tokens should have been out. If you try to find it in the chat, there's a lot here. If you would like to email me, if you have a question about the GNPS token or it didn't go through to you, please feel free to reach out to me.
I have a comment and a question. So I'll start with the comment. As someone who used beta versions of IDBac back in 2016, 2017 with Chase, I am just so impressed with the progress you guys have made. This is incredible. So major kudos to the whole team. I'm so excited to jump into this and implement into the science I'm doing now. And then the second question I had kind of was more, or the first question, I guess I should say, is towards the beginning, you showed the dry run feature, the yes or no, if you want your data to be uploaded. And I was curious, I guess the idea is that if it's not the really nice prim and proper data, you wouldn't want to upload into the database. and that's really just for searching? Or I'm just trying to understand like the main advantages or disadvantages when using that
Yeah,
toggle.
that is more of a sanity check. So ideally, what I like to do when I'm depositing strains into our database, I go through the analysis workflow first, and I can visualize my spectra again on that mirror plot spectra page. If I am happy with the spectra there, then I would maybe move on to depositions. But if you have
Gotcha.
already QC'd and you're relatively confident in your spectra, um, you can use, sorry, I want to go to that page that you're talking about. You can use the dry run to kind of do a sanity check to make sure that there are no scans that are empty. It
Mhm.
will essentially let you know if there's any errors. It will let you know if there's a problem with the metadata sheet instead of running the full deposition. It's not the end of the world if you submit and there's a problem. We can try to fix it, but it's easier to fix a problem before it's been deposited. So
Totally.
it's kind of a sanity check. One
Cool.
issue that I see happen quite frequently with metadata in particular is accidentally clicking one of these header columns and then adding an extra space or modifying these header columns even just a little bit. That can throw the whole system. So the dry run can just be a little bit of a fail safe for the user to feel a little more confident before proceeding.
Awesome. Okay, thanks. And then I missed the beginning because I was in another meeting, so I might have missed this. So sorry if I did. But
Mhm.
is there a documentation or anywhere where we can search what is currently in the library, the IDBac library, like what species or?
Yeah, so there
Okay.
is not currently documentation. I'm going to be working on some figures as this grows and just keep
Mhm.
updating it. But you can search it within idbac.org.
Oh,
If
perfect.
you
Okay.
want to, let's see, go down to full taxonomy, for example, you can toggle this to alphabetize. Oh, it moved over on me. You can alphabetize and then I got rid of it. Sorry.
Awesome. No,
You
no
can
worries.
search
This
it
is
that way.
perfect.
And
Mhm.
when you export it to, you can have an Excel doc that you can maybe look at the data a little bit easier than just on the interface. But
Awesome.
yeah,
Thank
thank
you
you for
so
your kind
much.
words. Appreciate it.
Yeah.
Okay, any more questions? All right. Well, if not, again, I want to thank you so much for your time, taking the time to be here with us today. I think Robbie at some point put my email in the comments. And again, please, we'd very much so appreciate if you fill out that exit survey so we can make things better moving forward. And yeah, I hope to speak with you all soon. Thanks again.